Automated Chemical Synthesizer by Alexander Chen and Griffin McCullough. Chemical synthesis is the process of performing chemical reactions.
Our goal is to create an automated chemical synthesizer that can mix two colored liquids together in a predetermined ratio.
A project applicable to the real world? Yes. In labs all over the world, automated chemical synthesizers are used to ease the process of creating chemical mixtures. This is especially true for the pharmaceutical industry, which relies on automated chemical synthesizers to keep up with demand, as the amount of chemical syntheses that needs to be done are unattainable by hand, but by using a machine, this project is exponentially faster, easier, and much more efficient. Our machine was based on the idea of continuous versus batch manufacturing processes. Batch processing consists of fabricating using bulk materials in batches. The downside of this is that it often causes a high amount of waste, and a batch cannot be started until another is finished. Continuous process is much more streamlined and consists of moving a unit through each step in production, resulting in a continuous flow of product. Continuous flow often results in less waste than batch process. An example of how continuous manufacturing is used in everyday life is two-part epoxy. The two different chemicals are mixed only as you push the syringe, creating the exact amount that you need. What this looks like in a chemical synthesizer is instead of having to mix each chemical in one at a time, you can simply press a button and have the mixture of your choice created before you in a continuous flow. Although our chemical synthesizer is not quite this streamlined, it operates under the same idea.
Our two colored mixtures are stored in syringes, which are then pumped by stepper motors. The liquids are pumped into a mixing/detection chamber, where using a blue and red LED and a light sensor, we can detect the concentration of each liquid. For example, when we shine blue light through our purple mixture, it passes through the blue liquid and gets absorbed by the red liquid. If the concentration of the red liquid is high, we get a low reading on our light sensor, because less light is transmitted through it. We can then take this reading, convert it into a concentration reading, which we can then use to trigger the stepper motors.
These phenomena are called the absorbance and visible light spectrums, and it's how we see color. Say a white light is shined on a green leaf. The leaf absorbs all the wavelengths that aren't green, and reflects the green wavelengths, and that is how we see colors. Although our machine cannot see color, it can read the wavelengths of them. The red dye absorbs the blue light, and the red light passes through, and vice versa. This can be done because the LDR has a sensitivity that varies with the wavelength of light that is applied to them. Our machine uses these principles and can calculate the concentration of either dye by using the amount of light that is passed through.
What would you do differently next time?
Next time we would spend less time on our calibration curves and our dilutions. This was a major time loss for us. With less time spent on these, we'd have more time to code our finished project.
What's next?
Instead of just mixing food coloring and water, we would want to move on to mixing actual chemicals. We could also up the complexity of our machine, like adding a third syringe or adding a heating element to break down solutes. With help from our mentors from SFU, we were able to complete this project. They showed us we needed a calibration curve in order to find the right starting concentration. A calibration curve is simply a range of values with known concentrations. It helped us find the lower limit and upper limit of our sensor. To create this calibration curve, we had to conduct a serial dilution. A serial dilution is a series of solutions, each diluted by the same dilution factor. For example, you would have 5 flasks, each containing 10 milliliters of water. You would then take a stock solution, mixture of known concentration, and add 10 milliliters of it into flask 1. You would then take 10 milliliters from flask 1 and add it to flask 2, and so on. With each transfer, the solution becomes more dilute by a constant dilution factor.